This lesson is on absorption and action spectra, and here are the objectives. Now, the wavelengths of visible light range from roughly 400 nanometers, that's blue-violet light, to 700 nanometers, and that's red light. So here, if you look at this x-axis, we've got violet-blue light here, so this would be sort of the 400 nanometer end of the spectrum, and here we've got red, and then going into the infrared, this is 700 nanometers. Longer wavelengths have less energy, and that's moving to the right on this axis here. An absorption spectrum shows the absorption versus wavelength for a particular pigment. So this on the y-axis is the absorption as a percent of the maximum. The main pigment that is in green plants is chlorophyll A. There are two types of chlorophyll that we'll talk about here, chlorophyll A and chlorophyll B. And chlorophyll A absorbs the blue and red lights best, so light at either end of the spectrum, but it doesn't absorb green light very well. It transmits and reflects green light. So if I were to draw an absorption spectrum for chlorophyll A, what I'm doing is I'm drawing how well that one pigment, chlorophyll A, absorbs light as a function of wavelength. And that's going to look something like this. Chlorophyll A absorbs purple or violet and blue light well, so it's going to have a peak like that, but it doesn't absorb green or yellow light well, so the absorption spectrum comes down like this, and then it absorbs red light relatively well as well, so there's another peak like that. So this is the absorption spectrum for chlorophyll A. Again, the absorption spectrum shows the absorption as a function of wavelength for a particular pigment. Now I said there's another kind of chlorophyll, there's chlorophyll B, and I'm going to draw a representative absorption spectrum for chlorophyll B here. It's also got a peak in the blue or the purple region, and then another peak over here in this sort of orange or orange-red region. You don't need to know the exact details of these shapes, but just understand that the absorption spectrum shows the absorption as a function of wavelength, and also understand that both chlorophyll A and chlorophyll B absorb purple and blue light and red light relatively well, but don't absorb green or yellow light. Because they don't absorb green or yellow light well at all, this light is reflected or transmitted and gets to our eyes, and that's why plants appear green, because they don't absorb the green light. There are other pigments called carotenoids that have a different shape absorption spectrum, so I'll draw a representation of that. They might look something a bit more like this. So there are carotenoids. Carotenoids are an example of an accessory pigment. Chlorophyll A is the main pigment in green plants, and the other pigments are called accessory pigments. They are pigments that help absorb other wavelengths. And so carotenoids or chlorophyll B are examples of accessory pigments. So once again, it's the light that is absorbed by the plant that is then used for photosynthesis, but it's the light that is reflected or transmitted by the plant that's what we see as the plant's color. So we see plants as green because they don't absorb green light very well, but plants are going to use the purple, blue, and the red light more for photosynthesis because that's what they're absorbing more. Now you might be wondering, how do we measure the absorption for an absorption spectrum? You use a machine called a spectrophotometer, and what you do with that is you put a solution of the pigment into a little beaker in the spectrophotometer, and then you shine a light through it. And you look at the light that comes out versus the light that went in. And any light that hasn't come out that went in was absorbed. And so that's how you can measure the absorption to figure out the absorption spectrum for a particular pigment. Now, the light that is absorbed, as we've said, can be used for photosynthesis. So an action spectrum shows the photosynthetic activity as a function of wavelength, and now we're looking at not just a single pigment, but for an intact chloroplast. So our x-axis is the same. We've got wavelength from purple-blue to red, so 400 nanometers to 700 nanometers, but now on the y-axis we've got photosynthetic activity, not absorption, as a percentage of maximum. And you could measure photosynthetic activity by a number of different ways, for example by carbon dioxide uptake, because carbon dioxide is an input to photosynthesis, or by oxygen production, because oxygen is a product of photosynthesis. And you can similarly draw a graph of photosynthetic activity versus wavelength, and it's going to look something like this. It looks like an envelope of all of the different pigments that we saw above, because, again, photosynthesis can only occur with light that's been absorbed. So if we are absorbing light in the purple, blue, and the red regions, and not in the green, yellow regions, then that's going to be reflected in the photosynthetic activity as a function of wavelength as well. We're going to get more photosynthesis at the two ends of the spectrum and less when we shine light in the middle here. Now, if you shine white light on a plant, it's getting all of the different spectra, and so it's going to use the light at the two ends of the spectra, the blue end and the red end, and it's going to reflect and transmit the light in the middle here, the green yellow, making it look like a green plant. So that's a look at absorption and action spectra and why plants look green.